Hello, my name is Julie Bowles. Today I will be speaking to you regarding recombinant adeno-associated virus vector-mediated gene therapy for the treatment of cystic fibrosis. I chose this topic for personal reasons. I have a friend who has fought with this disease since she was diagnosed at the age of 25. She is now 36, happily married with 2 children, and struggles to manage her condition every day. The average mortality age for someone living in the United States is 37, while some can live to their mid-50s. I'm passionate about finding a cure for this disease because her life and others like her will have years of their life taken. The research's importance stems from the fact that the most effective method to treat cystic fibrosis will be with gene therapy, since there are over 1,000 mutations and many patients develop side effects with the long-term use of pharmaceutical treatments. Gene therapy for lungs is difficult as they are covered in cells designed to keep foreign substances out. Patients with cystic fibrosis also have a thick layer of mucus lining the epithelial cells, which causes further complications. Specialized vectors will allow the correct protein to be uptaken by the cells. The most promising fact is that this gene therapy can be used for any individual with cystic fibrosis, no matter the type of mutation. Cystic fibrosis is an autosomal recessive disease that affects many Caucasians of European ancestry. A mutation in the cystic fibrosis transmembrane regulator, CFTR, a chloride channel that regulates water and salt transportation and produces a layer of fluid on the respiratory and intestinal tract, as well as several organs. When this fluid is absent, sticky mucosal secretions result in chronic infections and inflammation of the airways, obstruction of the GI tract, or difficulty with digestion, infertility in males, liver and lung disease. The majority of patients afflicted by this condition will die from respiratory failure produced as a result of chronic inflammation and bacterial infections. Gene therapy for the treatment of cystic fibrosis hinges on providing a regular copy of the CFTR DNA to airway epithelia by using a vector. The gene therapy can either be given through IV or by inhalation. The challenges of using gene therapy stem from the lung's function, as the epithelium has evolved a vast array of mechanisms for keeping microbes and other environmental factors out, which makes using a vector as a mechanism for delivery more challenging. Many different types of gene transfer agents can be used to treat cystic fibrosis. My presentation focuses on adeno-associated viruses, which will be covered in the next segment. One method is the Sendai virus, or SEV. It is highly effective due to the sialic acid and cholesterol receptors on the apical surface of the airway epithelial cells. A downside of this type of gene therapy is it is not well suited for long-term use, as it will result in absent gene expression. Since cystic fibrosis is a chronic condition, it is not a suitable technique for prolonged use. Lentiviruses are also highly efficient, and they have a unique ability to transfect both non-dividing and dividing cells, and are also able to be administered over prolonged periods, making it ideal for the treatment of cystic fibrosis. However, they lack receptors that recognize respiratory epithelia and naturally target hematopoietic cells. There are also non-viral approaches, such as cationic liposomes, a cationic lipid mixed with cholesterol and a helper lipid. While they have an increased chance of gene delivery, side effects resembling flu-like inflammation have been reported. My research project focuses on recombinant adeno-associated viruses, a vector-mediated gene therapy for the treatment of cystic fibrosis. AAV is a conventional gene therapy with low pathogenicity, and the vector can maintain transgene manifestation in non-dividing cells over many years. The vectors cannot replicate or merge into the host DNA, meaning they exist as an episome in transfected cells. AAV is a naturally defective for replication. Its genome consists of inverted terminal repeats, ITRs, and rapid cap proteins. 4 types of rep proteins are involved in replication, and 3 types of cap proteins are required for encapsulation and are produced via alternate splicing. Since AAV cannot propagate itself, it relies on co-infection with another helper virus. The most common is the adenovirus. For this gene therapy to commence, 3 components are required. First, a coding sequence of the CFTR gene is subcloned into a plasmid and bordered on both the 5 and 3 ends with ITRs. Second, an additional plasmid that has the rep and cap genes. And lastly, a method to provide the helper function associated with the adenovirus. The final particle will now contain the ITR, CFTR, ITR coding sequence that is encapsulated. It is known as a DNase resistant particle, or DRP, as a result of the DNA encapsulated within the viral particles is now fully formed and DNase resistant. The host cells will have the ability to have long-term expression of the recombinant protein due to the ITR, CFTR, ITR coding sequence being integrated randomly throughout the host cell. To determine if the cells are expressing the CFTR, immunofluorescence staining can be conducted. Often with CF, the gene can produce no protein, not enough of the protein, or it can be made incorrectly. Any of these defects will result in an array of symptoms that affect the lungs and other organs. The concept for restoring continuing function is to replace or repair the CFTR DNA sequence into the cell so that it can make the CFTR protein-producing fluid and prevent thick mucosal secretions, thereby eliminating the symptoms of the disease. Limitations of using the AAV repeatedly can create numerous problems. First, antibodies that have been created from previous usage can target virion proteins that eliminate the virus before it can infect host cells. Secondly, cytotoxic T lymphocytes can identify viral proteins that are expressed by the infected cells and eliminate them. One technique to overcome these problems is serotype switching. When using AAV normally, one serotype is used, and the second trial a second serotype will be used in order to avoid neutralizing antibodies that were created by the first serotype. The graph depicted shows the effect of a mucosylate agent N-acetylcysteine, NAC, on AAV transport in CF sputum and AAV transduction in BEAS2B cells. The BEAS2B non-tumorigenic lung epithelial cell line has been used as a lung epithelial model in various studies. And both studies depicted numerous particles tracking the AAV in CF sputum samples were left untreated or pre-treated with 5-NAC. The graphs show the distribution of the specific particle's mean square displacements over a period of 1 second. The data shows 5 sputum samples with an average of less than 900 AAV particles per sample. The results depict the mean cell GFP fluorescence measured by cytometry. In conclusion, cystic fibrosis is an autosomal recessive disease that results in chronic bacterial infections and inflammation. These symptoms are the leading cause of mortality in patients afflicted with the condition. AAV is a safe and conventional method for treating this condition, as it can be utilized in single and repeated dosing with no link to disease pathogenicity. It is a much more beneficial treatment, and there are over 1,000 mutations, which can be used with all of them. Pharmaceutical treatments are not effective in the long term, as immunity can develop and only treat symptoms. AAV can replace or repair the damaged CFTR gene, restoring function, and eliminating or reducing symptoms, which will ultimately lead to a cure. However, there are limitations of using this treatment that stem from antibodies that develop over time. Serotype switching is a remedy to alleviate the creation of antibodies, allowing the treatment to continue. The references are depicted, and the image credit slide as well. Thank you.